Thank Organizing Committee, Microbiology Society India, Udayana University Bali, Indonesia, Vidya Pratishthan College Baramati. So our risk is very high as a manufacturer. So that's why in pharmaceutical industry, stability testing of every dosage form is very important. Next slide, please. So it is important in the development of small molecule drug product. Yesterday also we saw in vaccine, right from development to launch in the market, and 12 months shelf life they have given. So that is also on the basis of their stability. Counterfeit drugs we have identified in many places. Title was remdesivir, but inside content was different. Next slide, please. Some visible problem that we can see by the naked eyes. That problems means missing of dosage form unit. Do you have experience? You have purchased one strip, one tablet is missing? Yes. Number of tablet missing. Sometimes empty. Sometimes discoloration happen because of micropuncture in aluminum foil. Next slide. And because of this loss of potency, 100% assay we cannot get. Loss of dissolution and loss of disintegration. Dissolution and disintegration means when you take the tablet inside the stomach and it is dissolved within 5 minutes or 10 minutes, that is known as disintegration. Dissolution means when paracetamol started its work, maybe within 30 minutes, within 60 minutes. Whenever you take paracetamol after fever, sometime within 30 minutes you can. Next. And big companies, they are using walk-in chamber and they are charging the product inside this chamber. They are monitoring after certain intervals. Next. So as per WHO, as per ICS, they have divided zone. Because worldwide temperatures are different. If you go in CIS country 2 to 8 degree or minus, Europe country always 20 to 25 or below that, India again all from FDA. And here 12 months. Next slide. And this is the requirement. Acceptance criteria, 5% potency loss from the initial assay value. This is significant change. This significant change should not be there when we launch the product. Should not be degrade. Always meet the specification. Next. This is the requirement. Those products are light sensitive. For that, photo stability studies is required. Yeah. Next. So we have done study on ibuprofen. Here, if you see, this is stored at 25 to 60% RH. This is 40 per degree and 75 RH. You can see the result are consistent. This is inconsistent result we got where in presence of UV and visible illumination. Next. You can see this is a control sample. Control sample means whenever we manufacture any batch and we supply in the market, we should keep one control sample 1 year after expiry. And it should be stable as per specification. And you can see this after this stability study, the appearance are completely different. So we cannot launch such type of product in the market. Next. That's why it is a costly activity. Such type of problems also we are getting during stability studies. Broken, completely melt, coating, completely disappear. Next. This also changes color from green to light. Here also you can see slightly change. Here original pack and different type of color is there in presence of light. Next. Then the color of the bottle, even color of the strips, showing changes. Next. Same photograph. Next. So before 1990s, as per FDA, the requirement was only temperature. 2000 temperature and humidity. And now temperature, humidity plus light. This is the mandatory requirement when we perform the stability studies.